today's video lecture, and in today's video lecture I'll be solving previous year's NEET questions and discussing the answers. And as you can see on your screen, it is for the chapter Human Reproduction, and we are in part 4 of the series. So if you haven't checked out part 1 to 3, then please do so. The links will be there in the description box below. Okay, so let's begin. Alright, so question number 1. What happens during fertilization in humans after many sperms reach close to the ovum? Okay, and this appeared in mains 2011. So let's go through our options. Secretions of acrosome helps one sperm enter cytoplasm of ovum through Zona Pellucida. Okay, all sperms except the one nearest to the ovum lose their tails. Okay, C. Cells of corona radiata trap all the sperms except one. D. Only two sperms nearest the ovum penetrate Zona Pellucida. So what happens during fertilization after many sperms reach close to the ovum? So secretions of acrosome helps one sperm enter cytoplasm of ovum through Zona Pellucida. That's what it is. So when many sperms, the answer should be A. Let's go to the discussion and I'll discuss this further detail. Now, during fertilization, many sperms enter the female genital tract, but very few sperms actually reach the ovum, right? So when they are in the genital tract, the female genital tract, there is capacitation of sperms, okay, which make them capable of penetrating the ova, okay. So now what happens is during fertilization, out of the few hundred capacitated sperms that surround the oocyte, only one sperm enters the oocyte. Okay, only one sperm enters the oocyte. So there are many sperms surrounding there, but only one manages to enter. So when one enters, then there are some changes that happen which avoid polyspermy. That is, they avoid many other sperms from entering at the same time. We don't want that. When one sperm enters, that's it, the job is done, right? Now, the enzymes present in the acrosome digest the corona radiata and Zona Pellucida and make their way into oocyte. So the acrosome of the sperm, which is present in the head of the sperm, contain many enzymes which help the sperm to penetrate into the ovum. Okay, and I will not discuss this in more detail over here in this question because I've already discussed the process of fertilization in another question where I've explained everything in detail. Okay, so but to answer your question, now if you can look at this diagram here, see, this is a normal oocyte that is there. Okay, now when sperm enters, there are many sperms, okay, entering, right? They manage to digest this corona radiata, but only one sperm will enter, okay, will enter via the Zona Pellucida. It will penetrate through the Zona Pellucida, okay, and then it will form the male pronucleus, and the female pronucleus is already present there in the oocyte, right? And that's when, you know, fertilization happens and the zygote is formed. Okay, so but like I said, to answer your question now over here, what happens during fertilization in humans after many sperms reach close to the ovum? Yeah, secretions of acrosome helps one sperm enter cytoplasm of ovum through Zona Pellucida. Okay, that's the right answer. Okay, let's move on to the next question. Okay, so question number 2. The second maturation division of the mammalian ovum occurs, and this appeared in 2010. So let's go through our options. A. Shortly after ovulation, before the ovum makes entry into the fallopian tube. B. Until after the ovum has been penetrated by a sperm. C. Until the nucleus of the sperm has fused with that of the ovum. D. In the Graafian follicle following the first maturation division. Okay. So your answer should be until after the ovum has been penetrated by a sperm. Okay. Here, second maturation division, they are talking about meiosis 2. Okay, now meiosis 2, let's go to the discussion, we'll discuss this in detail. Meiosis 2 occurs after ovulation when the oocyte is in the fallopian tube. But when the oocyte enters, as in meiosis 1 is completed beforehand, right, when before ovulation, so the haploid oocyte is formed. The meiosis 2 process begins, but it doesn't really complete. Okay, meiosis 2 is not completed by then. It enters the fallopian tube, the oocyte is there, but it is completed just before fertilization when the sperm enters the oocyte. Okay, so when the oocyte is there in the fallopian tube, okay, the meiosis 2 process has begun, but it is arrested at what? The metaphase 2 stage. Okay, it has, the process has begun, but it is not completed. So you can't say that the meiosis 2 process has occurred. It has not really occurred. It has started, but it has stopped in between. Okay, so it is not a complete process. Okay, it's still in that secondary oocyte stage, but it is stuck in between in the cell division cycle. So it is stopped at the metaphase 2 stage. Okay, now when, all right, so the oocyte is present there in the fallopian tube, we know that, right? So if a sperm comes in and it is penetrated into the ovum, the oocyte rather, okay, as soon as the head enters, the sperm head enters into the oocyte, the meiosis 2 process is completed to form a mature ovum. Okay, if there is no fertilization going to happen, the oocyte then gets discarded in the menstrual cycle. Okay, so the appropriate answer would be only meiosis 2 will occur only when there is a penetration by the sperm into the ovum. Okay, because before that meiosis 2 hasn't really completed, the whole process hasn't really completed, right? Only when that fertilization process is happening, only then it will complete. Okay, before the zygote is formed. So secondary oocyte then transforms into a mature ovum. So once the mature ovum is formed, then only the zygote will form. Okay, so the male pronucleus will, the female pronucleus will be there, and then the whole process happens to form a zygote. Okay, duplication of chromosome will be there and all that. So yeah, I guess the appropriate, if I go back to the question, the second maturation division of the mammalian ovum occurs. The appropriate answer would be until after the ovum has been penetrated by a sperm. Okay, but shortly after ovulation, before the ovum makes entry into the fallopian tube, well, the meiosis 2 process has begun then, but it's not completed, right? It stopped in between. Just like meiosis 1 that happens at puberty, at fetal stage, the process has begun, but it has not completed till after the female reaches puberty. Okay, so yeah, it is until after the ovum has been penetrated by sperm. This is the appropriate answer for this question. Okay, let's move on then. All right, so question number 3. Which one of the following statements about morula in humans is correct? Okay, this appeared in 2010. A, it has almost equal quantity of cytoplasm as an uncleaved zygote but much more DNA. B, it has far less cytoplasm as well as less DNA than it in an uncleaved zygote. Okay, C, it has more or less equal quantity of cytoplasm and DNA as in uncleaved zygote. And D, it has more cytoplasm and more DNA than an uncleaved zygote. Okay, so we're talking about morula. Okay, I hope you are aware of which stage is a morula. I have already explained this in the previous video, part 3, so please check that out. So yeah, in humans, it has almost equal quantity of cytoplasm as an uncleaved zygote, but much more DNA. Okay, so the appropriate answer is A. Answer is A. Let's go to the discussion. So during cleavage, the cells divide, but the cytoplasmic volume does not increase. Okay, so if you have a, this is Zona Pellucida, let's say you have this cleavage has happened. Okay, there is a nucleus over here, cleavage has happened. Okay, now this cleavage will happen over here. Okay, this cleavage will again happen over here, then there'll be more cells forming, then there'll be more cells. I have a diagram. Yeah. Yeah. So this cleavage is formed. Now you see that the division over here is not equal, right? You see the cells over here, one is bigger than the other, okay, during cleavage. So the cleavage that you can see over here, the division that is happening is not equal, right? Now over here you can see one is big and the other cell is small. So now it says the largest cell divides first. So this cell will divide first to form two more cells. Okay, like that there will be four cells, and like that you will find a form a morula. So you see over here, if you notice over here, the cell, the cytoplasm as such, the volume is still the same inside the zona pellucida. Okay, the volume is not changing, right? But the DNA content is more. Okay, the DNA is more, there are more number of cells, there's more nucleus present inside, but the cytoplasm as such is the same, the volume is the same, right? Because it is taken the whole, let's say if the cytoplasmic volume was 10, it will always remain 10 whether it's divided 5 plus 5 or 2 plus 2 plus 2 plus 2 5 times. Okay, the volume still comes down to 10, right? But the DNA content is more. This is 5 plus 5, so DNA content is less, but this DNA content will be more. Get that? There are more nucleus. Obviously, it has to maintain that diploid nature, right? Because this will eventually form into an embryo and then into a human being, into an infant. Okay, so to go back to our question, yeah, so during cleavage, the cells divide, but the cytoplasmic volume does not increase. The cells formed during cleavage decrease in size. So in a morula, or you've seen already, there's one big cell and there'll be a small cell, and this larger cell divides first, and then this goes on and on and on till a morula stage is formed, that is 16 to 64 cell stage, right? So in that you see that the outer cells are smaller than the inner cells, okay, and they are all unequal sizes, but the cytoplasmic volume is still the same, right? Cytoplasmic volume is still the same, but there will be cells of different sizes. Okay, and as the cleavage and all happens, as the cleavage happens, the cell begins to decrease in size. The cells formed, every cell is, you know, dividing and dividing and dividing. The cytoplasm is getting distributed, and the cells decrease in size. Okay, now the DNA content increases due to the replication of chromosome with each division, maintaining the diploid nature of each cell in the mitotic cell division. Okay, so it is a type of mitotic cell division that's happening in cleavage. It's not exactly like a normal mitotic division, but when we'll discuss this later on. I have a table I've made with the differences between cleavage and normal mitosis, but in this case I just wanted you to understand that DNA content is increasing there, but the cytoplasm as such is still the same. Okay, the cytoplasm as such is still the same. Okay, let's go back to our question, see if we've answered the question. So which one of the following statements about morula in humans is correct? So just remember this, if you get this in an exam, this is what it means. Okay, just understand the principle behind cleavage. So it has almost equal quantity of cytoplasm and as an uncleaved zygote, but much more DNA. Okay, so it has much more DNA, but it has equal quantity of cytoplasm. Clear? Okay, move on. Okay, so question number 4. In human female, the blastocyst, okay, and so on. This appeared in Mains 2010. A, forms placenta even before implantation. B, gets implanted into uterus 3 days after ovulation. C, gets nutrition from uterine endometrial secretion only after implantation. And D, gets implanted in endometrium by the trophoblast cells. Okay, so in human female, the blastocyst gets implanted in endometrium by the trophoblast cell. Okay, it gets nutrition from the uterine endometrial secretion, but let's say it does not happen only after implantation. So when the blastocyst stage is formed, it enters into the uterus region. It before the implantation also it needs some nutrition, right? So there are ways of sources by which the blastocyst gains nutrition, but it gets implanted into the endometrium by the trophoblast cells. Okay, so your correct answer is D. All right. So the discussion. So the transformation of morula into blastocyst occurs on the 4th to 5th day after fertilization. All right. Now, the developing blastocyst receives its nutrition partly from diffusions of uterine secretion and partly substances stored within the ovum. Okay, so when the blastocyst is still there before implantation, it is still receiving nutrition because it needs to survive, right? So it receives nutrition partly from diffusions of uterine secretion in the uterus also, and partly from substances stored within the ovum. Now, the thinning of Zona Pellucida occurs on the 4th day and disappears on the 5th day. The thinning is due to trypsin that causes dissolution of Zona Pellucida. So trypsin is an enzyme which is secreted and causes the thinning of Zona Pellucida. So before implantation, Zona Pellucida is a structure. It's very interesting to see. The Zona Pellucida is a structure which protects this blastocyst. Okay, and it does not allow implantation. With the Zona Pellucida, no implantation will happen. Okay, but the Zona Pellucida needs to disappear. That's called zona hatching. I've already discussed this previously, right? It's called zona hatching. So slowly by the 5th day, it's an approximate day, the Zona Pellucida begins to thin and disappears eventually. Okay, that's called zona hatching, and that is due to trypsin. All right, so now when the Zona Pellucida has gone, now the blastocyst is ready for attachment or implantation to the endometrium. So once the Zona Pellucida disappears, the trophoblast cells stick to the uterine endometrium and takes place on the 6th day after fertilization. So let's say it's interesting to note that once fertilization happens, within 1 week a woman is pregnant. That means implantation is already happened within 7 days, 6 to 7 days, an approximate. Okay, it is interesting to note that. So the trophoblast cells are actually very sticky in nature. Okay, so Zona Pellucida doesn't allow any attachment or implantation to happen. But once the Zona Pellucida disappears by the 5th or 6th day, then trophoblast cells, they have a very sticky nature and they attach themselves to the endometrium wall. Okay, and it takes place on the 6th day after fertilization. Now the polar trophoblast cells penetrate into the columnar uterine epithelium. Okay, I don't remember if I've explained this, but if you look at the blastocyst structure, if this is the trophoblast and these are the inner cell mass over here, this trophoblast, this region over here is called polar trophoblast. Sorry. Okay, this region over here is called polar trophoblast, and it's what attaches to the endometrium wall. Okay. They penetrate into the columnar uterine epithelium and between the mouths of the endometrial glands and provide passageway to the blastocyst. Okay. Now, secretion of proteolytic enzymes secreted by the polar trophoblast and the uterine epithelium causes erosion of endometrium. Okay, so there are some proteolytic enzymes which are secreted by the polar trophoblast and the uterine epithelium, which causes erosion, okay, of the endometrium. So now it has to move in, it has to erode into that endometrium wall. I think I have a diagram for this to show you. So if this is the endometrium wall, let's say if this is your blastocyst over here. Okay, it has to, this is the endometrium, okay, this whole thing, and it has to move in. Okay, so it has to somehow penetrate into the wall of the endometrium. Okay, this allows the blastocyst to burrow deeper and deeper till the whole structure is embedded within the thickness of the endometrium. So how does that happen? With the release of proteolytic enzymes. Now on the 8th day, when the blastocyst is partially embedded, the polar trophoblast cells divide into 2 layers. Okay, this is what happens. This is cytotrophoblast. It's also called Langhans layer. And so it is the inner layer of cells and is cuboidal to low columnar cells. Okay, so they are range from cuboidal to low columnar cells, and they have the ability to mitotically divide. Okay, another layer is called syncytial trophoblast. Okay, so there are two layers formed by the polar trophoblast cells. Okay, you know what the polar trophoblast cells are now? Let me show you again. So this is your blastocyst, I'm sorry, and this is your inner cell mass. Okay, let's say assume inner cell mass. Now two layers will be formed, and this, let's say this region over here is called the polar trophoblast. Okay, now from this region two layers are formed. Okay, there is a cytotrophoblast, so it is the inner layer of cells. Okay, let's use a different color. This is cytotrophoblast. Okay, let's say also called the Langhans layer. Okay, so this is the first layer, and it's another outermost layer. It's a multinucleated cell layer. Yeah, so it is the outer layer of multinucleated cells and don't have the ability to divide. Okay, so cytotrophoblast cells have the ability to divide, but syncytial trophoblast cells don't have the ability to divide, and they are formed from cytotrophoblast that have migrated to the periphery and fused. So you see this, let's say this is a cytotrophoblast. This orange layer is cyto. Okay, now syncytiotrophoblast would be I don't think you can see this, sorry about that. Yeah, use this color. Okay, the syncytiotrophoblast will be over here. Okay, so this is syncytiotrophoblast, and they emerge from the cytotrophoblast cells, okay, that have migrated to the periphery and fused. But the source is polar trophoblast cells. Okay, from there you have cytotrophoblast cells, and then after that you have syncytiotrophoblast cells. Okay, now these cells actually help in the implantation. Okay, so on the 9th day there is closure of the erosion of the uterine endometrium by the formation of coagulum of tissue fluid and debris that forms a fibrin plug. So once this whole structure, okay, this whole structure gets embedded into the endometrial wall. Now there's erosion happening, right? Now it doesn't want to leave an open space. That doesn't make sense, right? To leave an open space. So a fibrin is secreted to form a coagulum. Okay, so it forms a coagulum plug that covers the endometrial wall. On the 12th day, spaces begin to appear in the syncytial trophoblast cells which fuse to form lacunae. Now I already showed you, right, how the syncytial trophoblast looks. So if you have a syncytial tropho, let's say this again, I'll draw a photo or a diagram, sorry, of a polar trophoblast cell. This is the inner cell mass over here. So now you have cytotrophoblast cells over here formed over here, and syncytial trophoblast cells was here. Okay, syncytial trophoblast cells. Now what are lacunae? Lacunae are spaces. Okay, lacunae are spaces that are formed over here. Okay, there are spaces over here. Okay, they begin to appear by the 12th day. Spaces begin to appear in the syncytial trophoblast cells which fuse to form lacunae. Okay, so they fuse to form like bigger spaces. They're called lacunae. Now these cells erode the maternal capillaries which become congested and diluted to form sinusites. Now these cells over here, they erode the maternal capillaries, okay, and sinusites are passageways. So now what happens is when the syncytial trophoblast enters, lacunae are formed, and lacunae they interact with the sinusites of the maternal capillaries. Okay, let's say if they are like, see if you can see this clearly. Okay, so if this is the maternal capillaries, there are like sinusoids. Okay, they're like passageways, okay, which move into the passageways. Okay, they have to interact with each other, otherwise how will the embryo get nutrition, right? So this syncytial trophoblast, they erode the maternal capillaries, and so lacunae interact with the sinusoids that are formed. Just think of a sinusoids as passageways. Okay, they are like passageways, and they interact with each other. The lacunae and the sinusoids interact with each other. So this initiates a nutritive relationship between the fetus and mother. Okay, so now this whole process that I've just explained is implantation. Okay, I've just explained implantation to you in this particular question. I think I have a diagram. Yeah, I have a diagram also for this. So yeah, now you have the Zona Pellucida still over here, what I've just explained, right? And you have the trophoblast. So the Zona Pellucida lysis happens due to trypsin, and they have now, see over here, the polar trophoblast cells have already started eroding the wall, okay, the epithelium. Now they've already, now cytotrophoblast has formed here, sorry, okay, and syncytial trophoblast has formed over here, okay, with the help of polar trophoblast cells. Okay, polar trophoblast cells are usually present over here. Okay, over here, these are polar trophoblast cells. Okay, they are now eroding. Now they have entered that. Now fibrin, they enter the endometrial, the thickness of the endometrial wall, and so now of course there is an open space, so a fibrin plug is formed to cover that, right? Now once it enters, it has to form, that's not really shown over here very clearly, sorry, that the lacunae spaces are formed in the syncytial trophoblast cells, and they interact with the maternal capillary sinusoids. Okay, so syncytial trophoblasts help in that implantation. Okay, these cells help in the implantation. Okay, so if you see, by the 12th day or the 13th day, that interaction has been formed, okay, with the maternal capillaries, with the maternal blood capillaries, so that there is a nutritive relationship between the fetus and the mother, because the fetus needs, the embryo needs nutrition, right? So that gains it from the mother. So what I've just explained is actually the whole process of implantation. So this is what happens. If you like, you can note this down in your notebooks to help you understand. So in human female, the blastocyst gets implanted in the endometrium by the trophoblast cells. Okay, that is the appropriate answer. All right, I guess we can move on. Okay, so question number 5. A change in the amount of yolk and its distribution in the egg will affect, and this appeared in 2009. A, pattern of cleavage, B, number of blastomeres produced, C, fertilization, D, formation of zygote. Okay, so the change in the amount of yolk and its distribution in the egg will affect pattern of cleavage. Okay, so the answer is A. Change in the amount of yolk and distribution in the egg will, of course, it will affect the pattern of cleavage in an egg. This is actually pertaining to the animal world, not just human, because human egg doesn't, as in human zygote, doesn't have a yolk. Okay, so when cleavage happens, the cleavage is different from, let's say, a bird's egg or a reptile's egg or an amphibian's egg, right, or an insect's egg. Okay, so now their yolk distribution is different, and yolk plays a very important role, okay, in cleavage. Now usually in an egg, let's say, if the yolk is present here, okay, I'll just mark it with yellow color. So you know this is the yolk over here, right? And this is the cytoplasm area. This part is cytoplasm, and this is yolk. So when cleavage happens, yolk does not allow cleavage to happen, does not allow that cutting to happen. Whereas in this, where the nucleus is present, this area is easy for cleavage to take place. So when we are talking about human, there is no yolk, so it's easy for cleavage to happen. But when there's, let's say, a bird's egg or an amphibian's egg or a reptile's egg, there is some yolk present in that. So when we look at the animal world and the human, it is different. Animal world in the sense, the other classes, classes such as amphibia or reptilia or bird, all those classes which you will study in the animal kingdom, their yolk distribution is different. The amount of yolk present is different, right? But yolk plays an important role because wherever yolk is present, cleavage is a little difficult. Okay, cleavage is very slow, or it doesn't happen as smoothly as an area where there is no yolk, where there's cytoplasm present. So here, this region, cleavage will happen easily, but in this region it will be very difficult for cleavage to happen. So yes, the amount of yolk and its distribution in the egg will affect the pattern of cleavage. Simple, right? Just understand the function of yolk in the egg. When we look at humans, we don't really look at the yolk part because there's no yolk. They are elliptical eggs. But when you look at the other classes of the animal world, then things are a little different, right? So this is what I wanted you to understand when it comes to cleavage. Now there are different types of cleavage. There's holoblastic cleavage and there's meroblastic cleavage. Okay, the two types mainly. So in holoblastic cleavage, the segmentation lines pass through the entire egg. And this is what happens in humans. Okay, when there is alecithal eggs or there's microlecithal, very little moderate amount of egg in that. Okay, a moderate amount of yolk in that. Sorry, in microlecithal there's very little yolk. In mesolecithal there's a moderate amount of yolk. In there, holoblastic cleavage takes place where the segmentation lines passes through the egg. Okay, it's easier, okay, for the segmentation line to pass through the entire egg. Okay, and occurs in alecithal, microlecithal, mesolecithal. Examples are frog and human. The equal holoblastic occurs in amphioxus and marsupials, and unequal holoblastic occurs in lower fishes and amphibians. Okay, now meroblastic cleavage, the segmentation lines do not completely pass through the egg. Okay, and remains confined to a part of the egg. This is what I was trying to explain over here. Okay, so you have a huge amount of yolk. Okay, like macrolecithal egg with lots of yolk in it. Okay, like bird's eggs. There, then segmentation line will not pass through easily. Okay, like it will happen in a frog's egg or a human egg. Okay, so the segmentation lines do not completely pass through the egg and remains confined to a part of the egg. So it is found in megalecithal eggs as the yolk provides resistance to the cleavage. Okay, so it happens in birds, insects, reptiles. Okay, so then they may be superficial. The cleavage will be superficial, which occurs in centrolecithal eggs of insects, or discoidal as found in birds' eggs. And we'll come to that because I have got like good diagrams to explain what discoidal and the superficial cleavage is, which occurs in the meroblastic type of cleavage. Over here, yeah. So here what we are looking at is holoblastic cleavage. Okay, this complete cleavage. Now there are different types of eggs. Okay, isolecithal egg where the distribution of yolk is even. Okay, they're isolecithal eggs. So there you have radial pattern of cleavage. See, you see this radial, it forms a radial. Okay, and I just want you to understand, okay, for understanding this topic of cleavage, there is spiral cleavage. You can see how it forms a spiral. See this bilateral over here. There's rotational that happens in mammals. Okay, so this happens in isolecithal egg where the yolk is evenly distributed. Okay, now in mesolecithal, where there is a moderate vegetal yolk, there is a radial type of pattern of cleavage. Okay, it appears like this. Yeah, so another is meroblastic, which is incomplete cleavage. Like I told you, meroblastic, there is too much yolk in that particular egg. So let's say telolicital type of egg is present. Telolicital, where the egg is distributed in only one part of the whole egg. Or the yolk is distributed only one part of the egg. Okay, so you have this whole egg and the yolk is only present in one part of the egg. Okay, only at the bottom, only one section of the egg. So that is called telolicital type of egg. So in that, meroblastic cleavage will take place. But how will that meroblastic cleavage take place? And cephalopod, mollusk, and all bilateral pattern of cleavage will take place. In fish, reptiles, and birds, it will be discoidal. Okay, this forms a disc on top of that. Over here. Now, if the egg is centrolicital, where the yolk is present in the center of the egg, okay, now again meroblastic cleavage only will take place. Meroblastic cleavage, of course, it's incomplete cleavage, right? But how will the cleavage happen? It will be superficial. Okay, you can see it is all superficial over here, where there is centrolicital, where the yolk is present in the center of the egg. Okay, of course, in cases where there's like too much amount of egg present, those are called macrolicital eggs. So there's a whole table. I think I've discussed this in the previous part, or maybe it'll be there in this particular video. But to understand, this is only to help you understand what cleavage is and how it happens in the animal world, and how the amount of yolk and its distribution in the egg will have an effect on the cleavage. Okay, so if you ever get a question like this, you know that the amount of yolk and how yolk plays an important role, the position of the yolk in the egg and the amount of yolk in the egg will have an effect on the pattern of cleavage. Okay, now whether it will be a holoblastic cleavage or it will be a meroblastic cleavage. Okay, and based on that, once you are in the holoblastic cleavage section, you see where the yolk is present inside the egg. Isolicital egg, what kind of egg is it? Isolicital egg or some other egg? If I go back to that, or it's a mesolicital egg. Okay, see the isolicital or mesolicital, then be a holoblastic type cleavage where complete cleavage will take place. Okay, and if it's telolicital egg or centrolicital egg, that means where the yolk is present inside. Telolicital means only one portion of the egg, and centrolicital where the yolk is present in the center of the egg. Okay, so there meroblastic cleavage will take place. Okay, so there again pattern will change. Okay, so just to help you understand how yolk plays an important role, its distribution in the egg or the amount of yolk present in the egg will play a role in cleavage. If we go back to the question, a change in the amount of yolk and its distribution in the egg will affect the pattern of cleavage. Okay, so this clearly explains that, and yeah, this is the appropriate answer A. All right, so we can move on. Okay, so question number 6. Gray crescent in the area. Okay, and this appeared in 2005. A, at the point of entry of sperm into ovum. B, just opposite to the site of entry of sperm into ovum. C, at the animal pole. D, at the vegetal pole. So gray crescent area is an area that is present just opposite to the site of entry of the sperm into the ovum. Okay, now gray crescent area isn't usually seen in humans, but we see this in amphibians and other animals. But we look at amphibians, we look at frog in particular. Okay, in this case, so gray crescent area is the area opposite to the site of entry of sperm into the ovum. Okay, now let's understand what animal pole and what vegetal pole is. Okay, so an animal pole in an amphibian egg contains cytoplasm and nucleus, whereas a region containing yolk is called the vegetal pole. So let's say if this was the egg, so if this part contains the yolk, this part is the yolk, then what is this called? Vegetal pole, right? And if this part of the cytoplasm with the nucleus in it, this is called the animal pole. right? Now the sperm enters the animal pole side. Okay, so whenever fertilization has to take place, sperm enters over here. Okay, this yellow line over here is the sperm. Okay, entering the egg. Now when the sperm enters the egg, what happens? The sperm enters the animal pole side. This is obviously the animal pole side. It causes the cortical cytoplasm to rotate at 30 degrees. Sorry. So this cortical cytoplasm will rotate. Okay, it will turn this way. Okay, it will rotate at 30 degrees. Okay, whenever the sperm enters for fertilization, this cortical, and this happens, let's say this happens in frogs. Okay, this is what I'm talking about, in amphibians. So in the animal world, it is different from what happens in humans. Okay, so please understand this. And in this case, you can see the gray crescent area. Okay, so when the sperm enters the egg from the animal pole side, there is a change, a shift in the cytoplasm. Okay, the cortical cytoplasm over here, the cytoplasm that is at the edge of the egg cell, it rotates at 30 degrees. Now when that happens, the rotation exposes the cytoplasmic region inside the cell, which is gray in color. Okay, and this is called the gray crescent area. So when this shift happens, this cortical cytoplasm shift happens, this cortical cytoplasm shift happens, you will see a region of the cytoplasm inside. Okay, now think of it. Now what I'm showing you is actually 2D, but try to think of it as a 3D. You have a sphere, right? You have a sphere, and just imagine the sperm has entered and there's a shift. Okay, there's a shift in the cortical cytoplasm. A region of the cytoplasm will be exposed because of that. Okay, a region of the cytoplasm in the center will be exposed because of that, and it will be gray in color, and that region is called gray crescent area. Okay, this yeah, now this green area that you see here, this will be the gray crescent area. Okay, now this also part of the region of the cytoplasm, and it gets exposed. Okay, so the gray crescent area forms a future dorsal region, whereas the point of entry of sperm forms the ventral region. Okay, now it can be dorsal region, it can be it forms a dorsal region, and it also gives rise to the archenteron, the primitive gut, which we will come to later when we study about gastrulation in frogs. So what happens is the blastopore forms over here, and it eventually, I'll explain this a bit in detail later, how this area is important. It eventually gives rise to ectoderm, mesoderm, and endoderm, which then eventually forms the embryo, right? So for now, just understand that when the sperm enters this region, this gray crescent area region, is opposite to the entry of sperm. So wherever the sperm enters in the animal pole side, cortical cytoplasm shifts. There's a rotation of 30 degrees, and you see the gray region. Okay, you see that gray region on the cytoplasm, that's called the gray crescent area. Okay, and the gray crescent area eventually forms the future dorsal region. And the gastrula stage, the group of cells in the gray crescent area move into the embryo, forming a blastopore at its site, which I was just saying. At the site of entry in the gray crescent area is the dorsal lip. Okay, so a blastopore forms over here. Okay, and a region of cells, they're called bottle cells, which move in. Okay, they move in. I've got a nice diagram to show gastrulation of frogs. So they move in, and eventually they give rise to ectoderm, mesoderm, and endoderm. Okay, we'll come to that later. Okay, so this region plays an important role in the gastrulation. But yeah, for the sake of NEET, if they ever ask you this question, the gray crescent is the area that is just opposite to the site of entry of sperm into the ovum. Okay, it's not the animal pole or the vegetal pole, it is the area opposite. Okay, let's move on. Okay, question number 7. What is true for cleavage? This appeared in 2002. Change this. What is true for cleavage? Size of embryo increases, size of cells decrease, size of cells increase, size of embryo decreases. A size of embryo or decrease or increase happens much later. Obviously, the size of embryo increases, but at this stage, at the cleavage stage, uh, embryo really doesn't matter until differentiation of cells begin to happen, right, at the blastocyst stage. So what is true for cleavage? Then the size of cells decrease. Okay, option B. Yeah. So in cleavage, the size of cells decrease with each division. So you see this morula stage over here. This is the morula stage. You see there are cells of different shapes, right? Shapes and sizes. Sorry, different sizes over here. There are small sizes over here, the medium sizes, the big sizes. So cytoplasm volume remains the same. I'll just reiterate whatever I said. The cytoplasmic volume remains the same, right? So, but the size of cells decrease with each division. Sorry. So to answer your question, yeah, size of cells decrease. That would be the appropriate answer to this question. Question number 8. Blastopore is the pore of, and this appeared in 2000, is the pore of A. Archenteron, B. Blastocele, C. Coelom, and D. Alimentary canal. So I just discussed grey crescent area, right? So that's the region where the blastopore is formed, and that is the pore of Archenteron. Okay, the answer is A. So from the blastula stage, blastula stage is what? The cells formed around, and the blastocele is present in between. Okay, blastocele is present in between. Okay, this is the blastula stage, and we're talking about frogs. Okay, we're talking about amphibians. There is rearrangement of cells to form the 3 germ layers. Okay, so the blastopore is the first opening of the embryo, which will eventually become the mouth or the anus. So now it depends on whether which animal is it, because there are some in the animal world where the mouth develops first, and then there are some phylums where the anus develops first. Okay, so either the dorsal side or the ventral side will develop first. We don't know that. The space formed during that time is called the archenteron, which is actually the primitive gut. Okay, so protostome animals develop with the mouth first. Okay, they're flatworms, annelids, arthropods, mollusks. Sorry. So these are the phylums where the mouth develops first. And uterosome animals develop the anus first. Okay, they're echinoderms and chordates. Okay, so that blastopore region gives rise to the primitive gut, the archenteron gut, okay, which will eventually become the mouth. The blastopore is like the first opening. Yeah, it's the first opening of the embryo, which will eventually become the mouth or the anus. Okay. So as the bottle cells move in, it will give rise to a cavity inside, which actually forms the archenteron, the primitive gut. And have a diagram, like I said, it will eventually come. So please have patience. Question number 9. Fertilization. Okay, sorry, but before I move on, let me just make sure I've answered the question. So blastopore is the pore of archenteron. That is true. Okay, the blastopore gives rise to the primitive gut, the archenteron. Okay, and which will eventually then form the mouth or the anus, depending on which phylum it is. So there's some animals in some phylums where the mouth develops first, and there's some where the anus develops first. Okay, let's move on. Question number 9. Fertilization is a chemical substance produced from, this appeared in 97 and in 91. A. Polar bodies, B. Middle piece of sperm, C. Mature eggs, and D. Acrosome. So fertilization is a chemical substance that is formed from mature eggs. Okay, C. The answer is C. So fertilization is present in the ovum, and anti-fertilization on the sperm ensure that the sperm adheres to the ovum and penetrates it. Okay, so it is known to ensure that only the sperm and the ovum of the same species cause fertilization. This is very interesting. Okay, there are some peptides also present on the vitelline membrane, integrin peptides and disintegrin peptides on the sperm and the ovum, which also ensure that the same species of sperm is penetrating into the oocyte, okay, onto the ovum. And also in this case also fertilizin is a chemical that's present in the ovum, and anti-fertilizin on the sperm, which also ensures that the same species are interacting with each other. We don't want a dog and the human to fertilize each other, right? That will not happen. It'll never work out. But human to human, yes, it will work out, because they all contain the same kind of chemicals, okay, that interact with each other and allow for same species to interact, and so the sperm penetrates into the corresponding ovum. Okay. Sorry. So to answer your question, yeah, fertilizing is a chemical substance produced from mature eggs. Anti-fertilizin is present in the sperm. Okay. Right. Let's move on. Okay. So question number 10. In human beings, the eggs are, this appeared in 97. In human beings, the eggs are BA-lecithal. That's it. Okay, it's not mesolecithal, it's not microlecithal, it's not macrolecithal. In human beings, there is no yolk. Okay, so they're alecithal eggs. The answer is B. So in human beings, the eggs are alecithal, there's no yolk. Microlecithal eggs have, now this, remember this, okay, remember this list. Macrolecithal eggs have very little yolk, so sea urchin and starfish. Mesolecithal eggs contain moderate amount of yolk, and example is frog. Okay, so based on the amount of yolk and the distribution of the yolk, the pattern of cleavage will change. Okay, so this is where you need to apply your knowledge. Okay. So megalecithal or microlecithal egg contains very large amount of yolk. Okay, this example is reptiles and birds. All right, so you can note this list down. I remember I told you previously I had a list and I had made it, so you can note this list down. If they ever ask you a NEET, you can answer this question. Okay. But the question was pretty direct. In human beings, the eggs are alecithal. Just remember that. Okay. Okay, let's move on. Okay, so question number 11. In an egg, the type of cleavage is determined by, again, it's the same question. This appeared in 95. There are lots of questions related to cleavage. A, the amount of distribution of yolk, the number of egg membranes, the shape and size of the sperm, the size and location of the nucleus. So clearly, type of cleavage is determined by the amount and distribution of yolk. Okay, the answer is A. I've already discussed this, so it is the same answer. I will not go into much detail. All right, so we'll move. If you want to go back and look back at the discussion again, please do so. Feel free to do that. Okay, but I will not discuss this again. All right, let's move on. Okay, question number 12. What is true about cleavage in the fertilized egg in humans? It's appeared in 94. Again, a cleavage question. It starts while egg is in fallopian tube. B, it starts when the egg reaches uterus. C, it is neuroblastic. D, it is identical to the normal mitosis. So about humans, right? So it starts while the egg is in the fallopian tube. So fertilization happens in the ampulla of the fallopian tube. So once fertilization happens, cleavage begins. Okay, so the answer is A. So cleavage begins while the fertilized egg is still in the fallopian tube. Yeah, so if you want, you can go through or write this down, the difference between cleavage and what normal mitosis is. So cleavage occurs in zygote and parthenogenetic eggs, and this occurs in most of the body cells. Interphase is short, interphase of long duration, oxygen consumption is high, consumption is low, size of blastomeres decrease, size of daughter cells remains same, DNA synthesis fast and DNA synthesis slow, nuclear cytoplasmic ratio increases, the ratio remains same. Okay, so if you can note this down, this table, it will be helpful to understand cleavage and normal mitosis. What is the difference between the two? All right, easy. This was pretty simple. We'll move on. Alright, question number 13. Termination of gastrulation is indicated by, this appeared in 93. A. Obliteration of blastocele. B. Obliteration of archenteron. C. Closure of blastopore. And D. Closure of neural tube. Now what they're talking about is gastrulation in frogs, okay, or in other animals, but we'll talk about frogs in detail. Termination of gastrulation occurs when is indicated by the obliteration of blastocele. When the blastocele completely disappears, okay, that's when gastrulation is completed, where all the three germ layers are formed: the ectoderm, mesoderm, and endoderm. Okay, so the answer is A, and we'll look at the discussion in detail. So, in gastrulation of frog, the bottle cells involute into the embryo, displacing the animal pole towards the vegetal pole. Remember, I spoke about the gray crescent area, how the animal pole shifts. So, this is the continuation of that. Okay. So, once the fertilization happens, the gray crescent area is exposed. Okay. So, this is that bottle cells are present. There's some special type of cells. Okay. Bottle cells involute, they enter into the embryo, displacing the animal pole towards the vegetal pole. So the animal pole, this is animal pole, it keeps moving down. Okay, displaces. If the animal pole was over here, it moves down. Okay, shifts further. Okay, and simultaneously forming the three germ layers. So during the formation of the germ layers, the blastocele displaces, and by the end of the process is completely obliterated. So by the time all three germ layers are formed, the blastocele is completely gone, it has disappeared. Okay, and the three germ layers are formed. Yeah. So this is what I wanted to show you. So you see the blastocele over here, you see the animal pole here, you see the vegetal pole here. This is in frogs. Okay, this is what happens in frogs. Okay, so now we see the dorsal blastopore lip over here. Okay, there's an involution of bottle cells inside. Okay, and this animal pole has shifted down. Now when these bottle cells enter, what happens is archenteron forms here. Okay, so it gives rise to the, so blastopore gives rise to the primitive gut. It's called the archenteron, the gastrocele also. There's another name for it. Okay, so if they ever say gastrocele, that means they're talking about archenteron and the primitive gut. Okay, so they form this cavity-like structure. Okay, so it's called the archenteron. Okay, you see the animal pole has shifted, right? In the meantime, this mesoderm layers is forming. This dorsal mesoderm, this region over here, this red region over here is forming. Okay, now you see animal pole has shifted over here. This ectoderm is forming outside. Okay, this is the dorsal mesoderm. Okay, again it has become bigger. Now say blastocele has moved down. Right, blastocele has moved down. These eggs are also moving down. Blastocele is completely disappeared in this case. Right, animal pole has shifted further. Now this arrow means that the animal pole has shifted further down. Okay, now this is a little difficult to understand because this they have shown in 2D, but think of it as a spherical structure. Okay, it's a spherical structure. Okay, and these changes are happening. So the bottle cells enter, they give rise to a cavity. By then the animal pole is moving down. Okay, so it's giving rise, this bottle cells are giving rise to the mesoderm, and the ectoderm is also forming, and the yellow cells that you see here, these are forming, giving rise to the endoderm. Okay, so they are like pushing the blastocele further and further down till it completely disappears. See, you see this endoderm cells completely occupying the space of the blastocele. Okay, by then the ectoderm has formed, the archenteron has formed, the endoderm is also formed. Endoderm is completed. Ectoderm is formed, mesoderm is also formed, the archenteron is in between. Okay, this is the cavity over here. This is what you see as a final structure. So those bottle cells help in forming the whole structure, the three germinal layers, the embryo that is formed at the end. If you see now the final structure over here, you see the blastocele is completely gone. So basically, just for you to understand that when gastrulation happens, and this is in the case of frogs, there is complete disappearance of blastocele. Okay, by the end of gastrulation, when the endoderm cells completely push the blastocele away, that is, that space is occupied by endoderm now. Okay, there's ectoderm also forming, there's endoderm also that is formed, mesoderm is also formed. The endoderm, the yellow cells that you see here are completely pushing away, and this whole space is occupied by the archenteron. Now the whole cavity that you see here has become so big. It was small and slowly it becomes big and then big and pushes the blastocele away. Okay, so this is what's basically happening here. For NEET, I want you to understand this, so that if they ever ask you a question, you know how to answer. If they ever ask you in gastrulation of frogs, that what happens during that time. Okay, so blastopore is an area which allows for those bottle cells to, it is similar to the primitive streak in humans. Okay, we'll come to that later when gastrulation of humans, when we study that. But let's look at frogs. Okay, you see the animal pole has shifted and it has finally come down over here. Okay, it has finally come down over here, and this blastopore has formed over here finally. Okay, it has closed. Okay, it actually forms a yolk plug. The blastopore closes and forms a yolk plug. Okay, and you see the blastocele has completely gone. Okay, and it has been occupied by the endoderm cells, and there's a mesoderm, and then there's an ectoderm, and in between you have the primitive gut. Okay, now think of it as in the form of 3D when you see a spherical structure and see how that happens. Now what we see here is a 2D just for our convenience sake to understand. Okay, to understand how the cells are moving inside. Okay, so this is what I wanted to show you. But to answer your question, it's pretty simple. Termination of gastrulation, now you can see, because you visualize, you've seen the diagram, obliteration of blastocele happens. That's the termination of gastrulation. They're talking with respect to frogs. Okay, they're talking in that. So in the animal world, this is what happens. All right, let's move on. Okay, question number 14. In telolicital egg, the yolk is found, and we already discussed this. It's appeared in 93. So A all over the egg, B on one side, C both the sides, and D center. So telolicital egg yolk is found on one side. Okay, the answer is B. In isolicital egg, the yolk is uniformly distributed throughout the cytoplasm. In telolicital egg, the yolk is concentrated towards one pole, the vegetal pole. And centrolicital egg, the yolk is concentrated to the center of the egg with cytoplasm surrounding it. Okay, that's in insects. So you can also note this down again. Okay, the positioning of the yolk inside the egg. Okay, the amount of yolk and the distribution of yolk, yes, that matters in the pattern of cleavage. So now you understand, right? And to answer your question, the question was pretty direct. Yeah, in telolicital egg, the yolk is found on one side. All right. Okay, so question number 15. Acrosome reaction in sperm is triggered by, and this appeared in 93. A. Capacitation, B. Release of lysine, C. Influx of sodium, and D. Release of fertilizer. So acrosome reaction in sperm is triggered by capacitation. Answer is A. Let's go to the discussion. Now I've already discussed capacitation in detail previously, remember? And there was a point there where when there's influx of calcium ions, okay, it not only increases the movement of the sperm, but also it causes changes in the acrosome to make it easier for it to release enzymes. Okay, so yes, influx of calcium ions triggers that the acrosome to make it easy to release enzymes, but only when it is close to the ovum. That is when it touches the corona radiata and then zona pellucida. Okay, so the touching is very important. The touching causes this acrosome reaction to occur, but of course the influx of calcium ions at the time of capacitation makes it prepare the sperm to release those enzymes. So acrosome reaction is triggered by the influx of calcium ions at the time of capacitation. But actual reaction is happening only when the sperm touches the oocyte. Okay, so when sperms come in contact with the corona radiata of the oocyte, Perforations develop in the plasma membrane and the outer acrosomal membrane, and these release the enzymes. Okay, so if you look at the structure of the acrosome, I just want to show you the structure. I've explained the whole fertilization process in this. Yeah. So here you see you have the acrosome here, you have the inner acrosomal layer here, you have the outer acrosomal layer, and the whole this thing is the plasma membrane. Okay, and this is the nucleus membrane. Okay, so they have an acrosomal membrane, you have a plasma membrane, you have a nucleus membrane over here. Now perforations happen in the outer acrosomal region and the plasma membrane. You see this release of enzymes, this is what happens. Okay, this is what happens, and this is how the enzymes are released. Okay, these are perforations. This spaces that you see here, these are called perforations. Sorry. Let's go back to our question. Yeah, answer, sorry. So when this acrosome comes in contact with the corona radiata of the oocyte, the perforations then develop, okay, and they release the enzymes from the acrosome. Now what happens? Now when they release the enzymes, first corona radiata has to be removed, has to be moved out of the way, okay, to reach the oocyte, right? So corona radiata, what happens is hyaluronidase is an enzyme that is secreted from the acrosome, okay, digests the corona radiata cells. Okay, along with that there are some mucosal enzymes, and movement of the tail of the sperm also helps in the penetration. Okay, some mucosal enzymes are there in the fallopian tube as well as the movement of the sperm. Hyaluronidase also helps in the digestion of the corona radiata. So corona radiata is moved out of the way. Now next is zona pellucida. So the head attaches to the zona pellucida. Okay, now the glycoproteins on the sperm head bind with the receptors on the zona pellucida surface. Okay, ZP2 and ZP3. So there are two types of receptors present, ZP2 and ZP3, and the sperm head attaches to those receptors. Now what happens when the sperm attaches to those receptors? Acrosin is an enzyme which causes digestion of zona pellucida. ZP over here is zona pellucida, okay, for convenience sake. Acrosin is an enzyme that is secreted there which causes the digestion of zona pellucida around the sperm head. Okay, so if you have this zona pellucida, the sperm head is attached over here. Okay, acrosome causes the digestion of this region. Okay, this is gone. Oh, sorry, I can't do that, but because I'll lose the whole diagram. But just to understand, yes, no, that space is gone now. Okay, the space is gone now. All right. Now this attachment causes the changes in the zona pellucida and release of lysozymal enzymes by the oocyte cytoplasm. So now some structural changes happen. Now once that head has attached to the zona pellucida, some structural changes happen in the zona pellucida as well as some lysozymal enzymes are also released from the oocyte cytoplasm. Now the structural, this whole process is called zona reaction. Okay, please don't confuse this with zona hatching. Zona hatching is completely different, but this is called zona reaction. When the sperm head attaches to the receptors present in the zona pellucida, okay, there are some structural changes that happen around the zona pellucida region. Also, some lysozymal enzymes are also secreted from the oocyte cytoplasm. So now what that happens? Why is that happening? Okay, it is to prevent polyspermy. Okay, now once one sperm has penetrated, it has successfully reached the zona pellucida and attached itself, we don't want any other sperms, right? It has successfully attached itself onto the zona pellucida, we don't want any other boyfriends, right? We have one person and that's more than enough, okay, for it to cause fertilization. Okay, so this is to prevent polyspermy. Okay, this is to prevent any other sperms from entering. We don't want more than one sperms to enter into the oocyte and cause more confusion, right? So the structural changes that happen around the zona pellucida prevent polyspermy from happening, allow only one sperm to enter at one time. Okay, and this is called process is called zona reaction, and this is different from zona hatching. Okay, that comes later. Okay, So once zona reaction has happened, what happens? Now it has to cross the vitelline membrane. Okay, it has to cross that vitelline membrane. So when the sperm comes in contact with the oocyte, the plasma membranes fuse. Okay, it has touched, now it has gone past the zona pellucida and it has attached itself to the vitelline membrane. Now there are some peptides also present in the vitelline membrane which allow for the attachment. Okay, the plasma membrane of the sperm and the oocyte fuse. Okay, they fuse together. Okay, now this fusion occurs at the receptor sites that are species specific. Remember I spoke about fertilizing and anti-fertilizing, right? Fertilizing secreted from the eggs and the anti-fertilizing from the sperm, right? So there are, that is also species specific. Now there's an interesting thing that there are some receptor sites that are present there which is also species specific. Okay, these are disintegrin peptides which are released from the sperm, and vitelline membrane contains the integrin peptides. Okay, so we don't want a dog and a human or a crocodile and some other species to fertilize together, right? Nature doesn't want that. Okay, so nature has devised a method in such a way that only that same species uh, come together and fertilize. Okay, the sperm and the egg. Okay. So once the plasma membranes fuse, the plasma membrane fuse at the receptor sites, the integrin and the disintegrin peptide region. Okay. Now, once that happens, there's a calcium wave in oocyte. Okay. The contact with vitelline membrane causes depolarization in oocyte cytoplasm. Once this whole contact has happened, the plasma membranes fuse at that peptide site. Okay, we know for sure now the sperm is going to enter. It has reached to the oocyte cytoplasm. Okay, it has made its way successfully from by the corona radiata, by the zona pellucida, by the vitelline membrane, and into the oocyte cytoplasm. So this particular sperm has dared itself to enter and reach the oocyte cytoplasm, right? So most of the job is done. Now we are inside the oocyte cytoplasm. Now what happens when once the contact with vitelline membrane happens, there is a depolarization wave, the calcium wave in the oocyte cytoplasm. What happens is this causes meiosis 2 to be completed. So once its sperm head has made contact with the oocyte cytoplasm and caused depolarization of the oocyte cytoplasm, meiosis 2 is completed. Okay, and that's where the mature ovum is formed. So once that head has moved in, meiosis 2 is completed. Remember that this could be a possible NEET question. Okay. Now there's contact of cortical granules with the plasma membrane. Now some cortical granules are present around in the oocyte cytoplasm which come in contact with the plasma membrane of the oocyte. Okay, this is your oocyte. Cortical granules are present here. Okay, and they fuse with the plasma membrane of the oocyte. Okay, and they release some lysosomal enzymes from the granules that again cause and prevent polyspermy. So zona pellucida, zona reaction, and this is called cortical reaction. Both of them prevent polyspermy. Okay, zona reaction also happens. Some structural changes happen around the zona pellucida, which prevent from other sperms from entering. Again, cortical granules also fuse with the plasma membrane, release some lysosomal enzymes, which also prevent polyspermy. This is called cortical reaction. Okay, this is called cortical reaction. Now, changes in zona pellucida and oocyte plasma membrane prevent polyspermy. Already explained this. So remember zona reaction, and this is the cortical reaction. So the cortical reaction, what it does is it causes the vitelline to block and prevent polyspermy. Now, entry of sperm also causes some metabolic changes and help in the development of embryo. Okay, so the embryo, the developing embryo needs some food. So, okay, some nutrition, right? So there are some metabolic changes also that take place during that time. Okay. Once what happens? Now we know the sperm has made its way into that oocyte cytoplasm. It has allowed for cortical reaction, zona reaction to happen. Meiosis 2 is completed. Mature ovum is formed. The sperm head enter and the tail enter into the oocyte cytoplasm. Okay. So if this is the region of the sperm entering, the sperm enters. Okay, the sperm enters, meiosis 2 is completed by then, polysperm is prevented by Zona reaction, cortical reaction. We don't want any other sperms out of your way. Okay, we don't want you. What happens is the sperm head and tail enter and leave the plasma membrane behind. So what you see remaining here is the plasma membrane. Okay, now the plasma membrane has gone and now it is just there inside. Okay, the sperm head and tail. Now, the entry of sperm head triggers the completion of meiosis 2. I've already explained that, and reconstitution of chromosomes and forms the female pronucleus. Okay, so the female pronucleus is formed here. Meiosis 2 is completed, and the female pronucleus is ready for the fertilization to happen. Now, the sperm head makes a 180-degree rotation with its nucleus swollen and forms the male pronucleus. So the tail eventually disintegrates and this head, which contains the nucleus, forms a 180-degree turn and it forms the male pronucleus. Okay, so you have the female pronucleus, you have the male pronucleus, and now it's prepared for fertilization. Okay, so basically this is what's happening in the whole of fertilization. Okay, you have your corona radiata, you have your zona pellucida, you have zona reaction happening there, you have cortical reaction, the after it touches the vitelline membrane, the oocyte depolarization of oocyte cytoplasm happens, and finally the sperm is inside, excluding the plasma membrane. The whole sperm is inside. Female pronucleus is ready because meiosis 2 is completed. The head of the sperm head does a 180-degree rotation with the nucleus swollen to form the male pronucleus, and finally both of them are ready for fertilization. Okay, so to answer your question, this is what I wanted to discuss. Remember the first question we spoke about fertilization, right? So basically this is what it is. So acrosome reaction sperm is triggered by, the appropriate answer in this case is capacitation. Okay, so acrosome reaction happens, yes, the capacitation happens after the sperm has entered the female genital tract. Now during capacitation there is influx of calcium ions which then prepare the acrosome to release those enzymes, and when it touches the corona radiata of the oocyte, that's when the reaction begins, okay, to enter into the oocyte cytoplasm. Okay, so basically this is the whole process of fertilization. If you want, you can note it down in your notebooks to study as notes for your exam. I've tried to summarize as much as possible accurately so that it is easier for you to study. Okay, so let's go to the next question. Question number 16. Neuroblastic cleavage is a division which is, this appears in 92, here A horizontal, B partial or parietal, C total, and D spiral. So meroblastic cleavage is a division which is partial. Okay, it's not like holoplastic. We've already discussed this in detail, so I will not discuss this further. Oops, sorry. So meroblastic cleavage, the segmentation lines do not completely pass through the egg and remains confined to a part of the egg. Okay, they may be superficial or discoidal. If you want, you can go back to the previous question and see the discussion. I've explained this in detail. Okay, so let's move on then. Okay, so question number 17. Blastopore is, and this appears in 92, A, opening of neural tube, B, opening of gastrocele, C, future anterior end of embryo, and D, found in blastula. So blastopore is the opening of gastrocele. Okay, it forms the primitive gut, the archenteron. I've already discussed this when we studied gastrulation of frog. So from the blastula seed, there's rearrangement of cells to form the 3 germ layers. Okay, the blastopore is the first opening of the embryo, which will eventually become the mouth or the anus. The space formed during that time is called the archenteron. Okay, which is the primitive gut. It's also called as gastrocele. Clear? Yeah, so you have protostome type of animals, you have deuterosome type of animals based on the development, the phylum. So they're divided like that in the animal world. So please study animal kingdom to understand the classification of animals. Okay, we'll move on. Okay, so question number 18. During cleavage, what is true about cells? And this appears in 91. A, nucleocytoplasmic ratio remains unchanged. No. B, size does not increase. C, there's less consumption of oxygen. D, divisions like meiosis. I think we'll discuss cleavage in detail now a lot. So what is true about cells is the size does not increase. Okay, in fact it decreases. Cytoplasmic volume remains the same, but the cells decrease in size. Okay, so sorry, I'm sorry. So yeah, here I've already discussed the cleavage and the normal mitosis, the differences between the two. Okay, so I'll move on. We discussed cleavage in much detail already, so we'll move on. Question number 19. Freshly released human egg has, and this appeared in 91, A, 1 Y chromosome, B, 1 X chromosome, C, 2 X chromosome, D, 1 X chromosome, 1 Y chromosome. So freshly released human egg has, this is when it is haploid in nature, right? So it will have 1 X chromosome. Answer is B. Okay. So human ovum has an haploid number of chromosome, and the ovum contains only X type of chromosome. So in a diploid egg, if it's a female, she'll have XX chromosome, sex chromosome. Okay, that determines the gender. Okay, and if it's a male, it'll be X and Y, right? So when spermatogenesis happens, eventually the male forms a sperm. Either it will have X chromosome or Y chromosome, right? But the female doesn't have many options, right? So when oogenesis happens, it will always have X, irrespective of whatever it is. However oogenesis is happening, right, meiosis is happening, it will eventually come down to X. So the male can have either X or Y. Female will haploid, the egg, the ovum, that is, will always have an X type of chromosome. So when this ovum and the sperm meet and the fertilization happens, if the sperm has an X chromosome and the ovum X, it will form a female fetus, and if it is a male has carrying the Y chromosome in the sperm and fertilizes with the ovum, so X and Y will have a male fetus. If it's XX, then it is female. Okay, so basically the male is determining the gender, the sex of the fetus. Okay, it's not the female that does it, it's the male. I think we discussed this in the previous part also, part 3, where explained in detail. So basically this is what it is. Okay, let's move on then. Question number 20. Sperm and egg nuclei fuse due to, this appeared in 1990, A, base pairing of the DNA and RNA, B, formation of hydrogen bonds, C, mutual attraction due to difference in electrical charges, and D, attraction of their protoplasts. Okay, so the answer is actually D, and this is actually one of those vague questions where they just want to ask something, but I've tried to explain this in much detail, in the best accurate way possible. Yeah, let's see, if it makes sense to you. Now see, when we're talking about mammals, right, and I already spoke about fertilization in much detail, right? So we have a sperm and the egg which combine together. Especially in humans, there are certain peptides present or certain chemicals also released which make it species specific, right? So in the animal world, it is mostly like this, right? You can't have a dog and a human, sperm and egg fuse together and form some fetus, right? Doesn't make sense, it will never happen. But nature is designed in such a way, in the case of mammals, that there are receptors present on the egg and sperm which allow gamete recognition for a particular species. So there are certain chemicals that are released, okay, from both sides. But in flowering plants, okay, sexual reproduction in flowering plants consists of an immotile sperm. So there's a pollen tube that is carrying the sperm. Okay, it is not like in males where sperm moves on its own and reaches the egg. No, there's a pollen tube which carries the sperm and drops it in front of the ovule, okay, with the help of a pollen tube. But obviously, yes, that makes it, easier for the sperm and the ovule to meet. But let's not just assume that maybe, species specific chemicals or peptides are not present there. Okay, because there's not much research done in this field. Okay, where there are species specific chemicals or attractants, which attract the sperm and the egg of that flowering plant to fertilize. Okay, so it can be assumed that there are some chemical attractants that bring the sperm and the ovule together and cause plasmogamy. Plasmogamy is actually the fusion of the protoplasm. Okay, it's not really the protoplast. Protoplast is different, where the cell wall is removed. But here, what happens is that causes plasmogamy. Okay, because there was a research done, and I found this article where they had done an experiment and they realized that the nuclei of the sperm had shifted. Okay, which then saw that there was plasmogamy that happened in the flowering plant. So yeah, to answer your question, yes, some attraction is possible that is happening between the sperm and the ovule. Okay, in this case, to answer your question, the others A, B and C don't make sense, but there could be some attraction of their protoplast. That's what I assume. And yeah, it's one of those vague questions which we have to deal with. Okay, so also you can use the process of elimination and find the appropriate answer for this question. All right, so yeah, let's move on then. Okay, so question number 21. Cells become variable in morphology and function in different regions of the embryo. This process is it's appeared in 1989. Differentiation, B, metamorphosis, C, organization, and D, rearrangement. So cells become variable in morphology and function in different regions of the embryo. The process is differentiation. Okay, there's differentiation of cells which make them variable. So the answer is A. So in the early stages of development itself, the blastocyst transforms into 3 germinal layer embryo. Remember, the trophoblast cells also then divide into the cytotrophoblast and syncytiotrophoblasts. Already differentiation is happening there itself. Then the embryonic disc is formed from the inner cell mass, and that again eventually gives rise to the ectoderm, endoderm and mesoderm. Okay, so they all have specific functions. So all this is differentiation happening. So the inner cell mass and the trophoblast differentiate into ectoderm, mesoderm and endoderm. Oh, I'm sorry. So to answer your question, cells become variable in morphology and function in different regions of the embryo. The process is differentiation of cells. Alright, so I guess this is the last question. And we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture and hope you've learned something new today. So please don't forget to like, share and subscribe to MG NEET Biology, and I'm available on Twitter, Facebook and Instagram. So links will be there in the description box below. You can follow me there for more updates, and I will see you in the next video lecture. Right, so bye.